Hi, everybody. So welcome back to the session. So let's start. Wow, session number 2. Okay. So the speaker of this first speaker of this session is Marcello Rosa, Monash University, Australia. So the title of his just talk is "Understanding the Human Visual System Through the Study of Non-Human Primates." So please welcome Marcello.
Hello. So let me just see to skip to the next slide. I can just, okay. All right, so thanks organizers for bringing me here today. And today I only have one disclaimer, is that in this talk there's no neuroimaging. But having said that, I will follow Anna in emphasizing thalamocortical circuits as an important part of the neuroanatomy that we have to pay attention to. So the first part of this talk is introducing an animal model that my laboratory has developed over a number of years, specifically for studies of types of lesions that are unique to human and primates and cannot be studied as well in other animal models. But then I will also touch upon recent results on the plasticity of the visual system and at a much more cellular level than neuroimaging. So the first question is, why do we bother studying primates at all? And the first thing I want to say is that you have to be very selective and look at the question, that because for various reasons, you want to use the animal model that is simplest and can facilitate your research, but at the same time replicates the essential features of the system or the question that you're studying. So the first reason why you want to study non-human primates is because, well, at the cellular level, in developmental level, they are different. So if we compare, for example, the brains of two animals with the same body size, or so the same, for example, the mouse lemur there and the rat, the mouse lemur brain is much larger. And these are actually drawn to the same scale. In fact, in order to get an animal that has the brain about the same size, not quite as a macaque monkey, you would have to go to a capybara, which is 40 times bigger. So clearly, this demonstrates that there are some things that are special and different about the primate brain. And one of the ones that we care most about is the fact that the cerebral cortex has subdivisions that are simply not replicated. So for example, this is a very simplified slide from Leah Krubitzer. It shows what the cerebral cortex would look like if all the gyri and sulci were unfolded and just laid flat on a dish. So the important thing that I want to show you here is that if you compare, the areas are color-coded, so corresponding areas are in the same color, but the gist is that there's a lot more white between the colored parts. So these are what we call the association cortex. This expands in proportion to the primate brain size, but in a predictable manner. So it's not like, for example, you would expect that the human brain has additional areas because these were developed for something. The evidence actually points that there's a developmental program that as your brain becomes larger, more cells are added, and specifically in the regions between those colored bits, and those parts become used for things that we care and value about. So now The animal model that I actually use is called the marmoset monkey. It's the size of a rat. It's about 300, 400 grams. It looks like that. And it's slightly more separated from humans in evolutionary terms. So for example, if we look at the evolutionary tree of primates, the macaque monkey, which is usually is still the most used animal model in research, was separated from human about 25 million years ago, whereas marmosets about 40 million years ago. But most of the differences that we could trace are related to the brain size itself and the separation and volume of the association areas. Having said that, there are excellent models for the study of the visual system, which is what I care most about in my research, because they share specific features with humans that you do not have, for example, in mice and rats. And one of them is going to be the topic of the lecture today. So to give you some idea, drawn at the same size, marmoset brain there, macaque monkey there, you can see at first glance that it's smaller, but also that the brain is much less convoluted. Therefore, this in itself is an advantage. For example, if you want to do multi-electrode arrays across areas, optical imaging that my laboratory also does, or registration to templates with a much higher level of precision with respect to site architectural areas. And the final advantage of using the marmoset is the lifespan. Now, for people who use mice, I use this slide and they get horrified that you actually have to wait 1.5 years for the animal to become adult. But in reality, this is as good as it gets if you care to study a primate brain, if your question justifies. So this is pretty much aligned with humans, so they become sexually mature at about 15 months, and the whole lifespan, 10 years is already a very old marmoset. 16 and 17 is sort of the record in colony protection environments. So what this gives us is the chance to do developmental studies across the lifespan within the duration of 2 or 3 project grants, which in the macaque monkey, it wouldn't be possible. Now, I mentioned that the marmoset has a very developed visual system, and one of the things is the visual cortex. So this is a lateral view of the brain and a medial view of the brain. Frontal lobe here, occipital lobe there. And you can see that about half of the marmoset brain is devoted to visual cortex. And most of this is actually, by the way, not an artificially inflated brain, as you see in human neuroimaging. This is actually what it looks like. So it only has the calcarine sulcus on the medial surface. It has the lateral sulcus where the auditory cortex is. But all of the visual cortex is actually exposed on the surface. And we have, I'm not going to talk much about this today, but we have used this to our advantage in electrode array and optical imaging and other types of studies. So it has the same basic subdivisions of the human visual system. The proportions and sizes are different. Today, in particular, I'm going to be talking about the primary visual cortex and the middle temporal area, also named area MT. And the topic of the lecture is what happens when you lose V1. So going back, if you have damage, which is relatively common to this part of the brain, people become blind. And this is actually not the way it goes with most animals. Because the thalamocortical connectivity in primates is such that the lateral geniculate nucleus, which, as you heard from Anna, is the primary nucleus for conveying vision to the cortex, if the primary visual cortex is damaged, where all this projection focuses, people become blind, even if all these other areas are still intact. Now, for example, if you do the same to a rat or to a mouse and just destroy the primary visual cortex, their behavior, they lose a little bit of visual acuity, but they can still live, for most intents and purposes, a happy life. So this is different because the connectivity between the thalamus and the cortex is more widespread in non-primates. For example, the lateral geniculate nucleus, if it loses its main target in V1, it still has enough projections going directly to V2, V3, V4, and other areas, or homologs of these areas, that can still keep the function going. But in humans and primates, the story is different. And this is actually one of the people have known that damage to the occipital lobe causes blindness for a long time, but it was not until World War I, or actually the Sino-Japanese War in the beginning of the 20th century, that with the event of having high-speed projectiles going through the skull, that people managed to precisely say, not only, yes, blindness comes from damage of the primary visual area V1, but also to map the relationship between where the damage was and what in the visual field the damage was. For example, if the damage is right at the back of your brain, you have damage to central vision. If the damage goes from one side to the other, damages the calcarine sulcus but spares the occipital lobe, you have damage to peripheral vision but sparing of central vision, and so on. So, but not everything is lost. So sometimes the patients still had some perception of motion. So they would describe this as being, for example, if someone had closed my eyes and was waving the hand in front, I could say something was moving there, but I couldn't say what it was. So this was called the Redox effect, and because the knowledge of damage to V1 causing blindness was so ingrained, this was ignored for many years. And then many years later, Laura Weisskranz in Oxford wrote a monography called Blind Sight and showed that not only you have this sort of vague conscious vision, but there's also a number of other visual capacities that remain, that if people are put in situations where they have to guess, if they don't have the conscious perception, but they can still guess sort of what's going on. So I think this is an interesting problem to study in primates because of the uniqueness of the circuits, but also, it's an interesting phenomenon on its own, but it's also a problem without a clinical solution. People, even 100 years and 120 years later, they're still getting blind, and the recovery is limited, and so understanding the circuit can give us clues of where to target the treatment or the ways. So, as I mentioned, the initial idea is that in the normal animal, the retina projects to the lateral geniculate nucleus, to the primary visual area, and V1 projects to the other areas. If the damage to V1, there's damage to V1, this circuit's interrupted, but the question is, what's bringing that other information? What explains blind sight and the conscious perception of motion that survives? And this is what we've been addressing. Now, we've done this with lesions to the V1 in many marmosets. This is an example registered to a template. The extents of the lesions are in gray. So this is the cortex, frontal lobe here, occipital lobe here, primary visual cortex is the big one here. And we managed, because of the geometry of the marmoset brain is simplified, we can do this lesion fairly consistently in a large number of animals. Now, the big debate, I will skip this slide briefly, just mention that the big debate in this field is whether blind sight and conscious motion perception come from a circuit involving the superior colliculus and pulvinar or something else. So this is shown here in this textbook diagram. So most of the visual information goes to the LGN, to V1 and other areas, but there's smaller, so to speak, circuits that go to the superior colliculus and pulvinar. And very influential research in the 1990s and 80s has pointed to the importance of the superior colliculus and pulvinar. But nowadays, the field has evolved, and there is evidence that there's different aspects to blind sight. For example, projections across the amygdala, explaining affective blind sight. And specifically today, I'm going to try to talk about the motion perception. So lateral geniculate nucleus has been dismissed for many years as a potential conveyor of blind sight and residual vision. So the main reason for that is that you lose most of the cells there. So this is a picture of the lateral geniculate nucleus on the intact size and the affected side. Those lesions are unilateral. So you can see that there's shrinkage and cellular loss. There's a very well-defined region here where cells are lost. That's called, we could refer this as the lesion projection zone. Now, this sort of and this happens very quickly. So this is cellular loss in the LGN, neuronal density, at different times after the lesion. So one of the questions we addressed was, is the survival time different? It makes a difference to how many cells survive. And the answer is, this happens within a month. You can see those points here are all the same except for this one. This one is the cellular density at 2 weeks after the lesion. And after that, if you look at 1 month, it's already at a steady state. If it goes over 12 months, there's no further cellular loss. So there's 2 ways of looking at that. First of all is, oh my God, most cells are dying. There's nothing we can do because it's too quick. The patient's still going to be in the ICU. Another way is to say, hey, there's about 30% of the cells that survive. What we can do with those? Right? So we've been focusing on those cells, the ones that survive. So in order to, first of all, I'm going to be talking to this in front of you, point of view of anatomy, and then I'm going to talk a bit of physiology. So if you, I showed to you the middle temporal area, area MT, which has been one of the areas where focus on research in blind sight has been given, essentially because MT is, for the lack of a better description, the cortical motion center. It's where direction of motion of objects is decoded and this information is sent to other areas. And because motion is one of the very few things that survive, people have focused in area MT, and there's a large literature showing that there's rearrangement of white matter tracts to area MT, that the amount of fMRI activation in MT, it correlates with how much the person can actually perceive, and so on. But we wanted to see, and one of the things is that area MT receives a sparse but consistent projection from the lateral geniculate nucleus. So the LGN, 98% of the cells in the LGN project to V1, but there's a small population that projects to MT. But when I say small, this is really small. This is sections through the lateral geniculate nucleus. The lower layers or magnocellular layers are shown in light gray, the parvocellular layers in dark gray, and each of those dots is one neuron that projects your area MT. So you can imagine that if you had made this injection in V1, all of this would be cell, sort of thousands of cells here. So if you make a big tracer injection in area MT, and these are retrograde tracers, so these are substances that you inject in the target area, they are carried backwards to the cell body. So you're revealing the neurons that form a connection to a specific target. And you can see that there's very few of those, and they are located exclusively in the gaps between the main layers. So for example, parvocellular, magnocellular, there's a few cells here, a few cells there, a few cells in what's called the K layer, which is the bottom layer of the LGN. But these are well defined and very sparse. Now, what we found was that if you make a lesion of U1 and wait for more than a year, this situation changes a bit. First of all, you see a lot more of those cells, but also they are located in the column that spans most of the LGN layers. And specifically, they are located in the lesion projection zone, which is, I showed you a few slides ago, that region of degeneration. So that region that most cells die after V1 lesion is specifically where you find those. And there's nothing sort of discrete about those cells. So this is an actual picture of the lateral geniculate nucleus under fluorescence microscopy. And you can see those huge neurons labeled in blue and green there, which are the individual cells that are labeled by the tracer. Two things, two take-home messages. First of all, they are only located in this sort of darker region in the middle, which is the lesion projection zone. Those regions here still have connections to the remaining parts of V1, but also they are located across the spectrum of potential layers. So, and the second thing is that these cells are big enough that you can even see them labeled at this magnification. So something strange is going on there. Now, among these strange things that happen, in the normal marmoset, as in humans, as in macaque monkeys, there's a very specific expression of calcium-binding proteins. Our question was actually, which cells are those? Do they identify as corneocellular layer or cells in those gaps, or they are in the main layers, parvo and magnocellular? So in the normal primate, you have a very specific segregation. The magnocellular and parvocellular layers, the layers that send most of the projections to V1, only express the calcium-binding protein parvalbumin. Those gaps, or these smaller layers, which are shown in green here, they express calbindin. So there's mutual exclusivity. You have two populations of cells. If you wait after the lesion, the story changes in that most cells in the M and P layers start expressing, they continue expressing parvalbumin, but they started also expressing calbindin. So many of the cells there have a sort of orange and yellow. So this specificity is lost. The cells in the K layer, most of them still remain true to their neurochemical identity. They still express calbindin. But this gives us this, if you want to ask what are those cells, you cannot use the classical criteria. What we were hoping was that there will be red or green. So are these MNP or they are K cells? So, but if you take the criterion that, okay, now the MNP layers are expressing both, so this is showing control. You have, in a control animal, no lesion, or the opposite hemisphere, in fact, of the same animal. You have cells that are either calbindin positive or they have no marker in the, and so calbindin expression in the, and then in the after neonatal lesion, you start seeing cells that express both. These are, okay, I forgot to say one thing. In this slide, what I'm showing you are those specific neurons that form the connection to area MT. In the same animal, they were labeled with a tracer after injection in area MT. So the question here is, what are those cells that form the connection to area MT? In a control animal, they either only express calbindin because they are in the K layers, or they express no marker. After neonatal or adult lesions, you have this population of cells that expresses both. So there's indication there that those cells in addition to their geographical position in the layers of the LGN, they might be modified M and or K. Specifically, most of them actually look like M cells, including the fact they are very large cells. So there is potential there. There's active plasticity that can help in after, and there's a population of surviving cells that if we can find how to improve them or to boost their activity, they are potential therapeutical target for making the most of what survives after V1 lesion. So this is the summary of this part of the talk. In my last 5 minutes, I will just ask a different question, which is, okay, you have those cells, they are in the correct location in the LGN, they form connections to area MT, but are they actually signaling any visual information that is relevant for a patient? So specifically, are they signaling information from within the blind sector, which is called the scotoma, which is the region that corresponds to the damage in V1. So in order to do that, we did electrophysiological recordings in the marmoset. So the experiment went like that. So this is a lesion. You can see the calcarine sulcus is still present. So as in those lesions that I showed before, this causes a scotoma. In this case, the scotoma or regional blindness can be mapped directly in each individual animal. So this is a perimeter showing half of the visual field from fovea to 90 degrees periphery. We record receptive fields all around the edge of the lesion, and what's left in the middle is the size of the blind region. So this we are looking at, is the LGN, the lateral genicular nucleus, still sending information specifically about that region? And the answer is yes. So as you go down with an electrode, recording the receptive fields of single neurons in the lateral geniculate nucleus. So here's a diagram, for example, of the LGN with the different layers. So these are for ipsi, contra ipsi, or contra ipsi, I forgot. They are dominated by one of the eyes. I forgot exactly. So yeah, well, there you go. Ipsi is orange and yellow. Contralateral eye is blue tones, right? So, and if you go in the normal marmoset or normal primate, in fact, and go with an electrode down the layers of the LGN, the pattern is that the receptive fields start near the fovea and move towards the periphery and into the upper visual field. And you can see that there are many white sites, so no neuron recorded, but when you do find a neuron, they have receptive fields inside the blind region. So those neurons, these receptive fields were mapped with electrophysiological techniques. So the key take-home message here is that not only these many months after V1 lesions, you have the receptive fields capable of signaling information from inside the blind region, but also the general topography of the lateral geniculate nucleus is kept more or less what you would expect. So those cells that do survive seem to be still doing a good job. And I'll show you more evidence of that. So we see the same types of neuronal responses that you see in a normal lateral geniculate nucleus. So as in the previous slide, I forgot to mention that, but the pattern of eye dominance between ipsilateral and contralateral eye was preserved, but also you have the responses that continue to be very strong, and they have on-off responses, off only, and so on. Irrespective, just for information, because there's slight differences between geriatric animals, the WG, for example, and the young ones and the adult ones. So, but in reality, we found very little difference depending on when the lesion happened. The only thing that was observed that was significant also was that some of the receptive fields were significantly larger than you would expect. The ones that are shown in color here are the ones that were above the expected level of significance for the layer where they were recorded. So it does seem like there's also some rearrangement at the cellular level. We're still investigating if this means that the dendrites of the cells are extending further or what are the mechanisms. And the final message is that the response strength and the response latency were indistinguishable from those observed in the contralateral side or in normal animals. One important point here, though, is that those latencies were about 30 milliseconds, so it's the time that takes for the cell to respond to a flash of visual or presented in the visual field. But these are shorter than what you observe in area MT of the same animal. So this argues that the LGN is actually sending information rather than being activated by feedback connections from area MT. The timing is such. And that brings me to the end of my lecture. So the take-home messages are the LGN neurons that survive V1 lesion continue to represent the visual field inside the scotoma. They have slightly larger receptive fields, many of the cells, but their responses to visual stimuli resemble those in non-damaged brains. The response latency argues against this activation inside the scotoma coming from feedback from area MT. So they seem to be viable candidates to mediate some aspects of residual visual ability, follow V1 lesions. Why I think the final take-home message is that there are some conditions that require a primate model in order to investigate, because the anatomy in this case is so specific and unique to primates that you have to have an animal model that replicates that specificity and that complexity. And in this case, the marmoset seems to be the simplest primate that still preserves that, those characteristics that you want in order to make studies that are relevant for humans. Right?
Thank you very much. So now it's time for questions. If you have a question, please come to the microphone.
Hi, that was a really interesting talk. I have a question of curiosity, which is, in normally aging animals, are there any differences in these circuits in terms of?
Okay, we've done research on very old animals, and two things came out. One is that the severity of the cellular loss in the LGN is significantly higher if the animal receives the lesion over 10 years old as compared to a young adult, right? So there's definitely an effect of aging on the susceptibility of neurons in the lateral geniculate nucleus to withstand and continue signaling after that. In the physiological studies, we found mild differences across groups. For example, the latency could be like 5% larger, but it's difficult to explain if this was because more cells were lost, and the ones that were lost were the ones that tended to be the highest latencies or so on. So the reality is that we were surprised, in fact, by how little difference the age makes. Age at which the lesion was sustained for those measures that we got.
Thanks, Marcello. That was a really great talk and one of my favorite topics of blindsight. My question actually kind of follows up from the time point of when the lesion has happened, because of course there's thoughts about the LGN pathway, but there's been also evidence that the Pulvinar pathway might enable blind sight or residual vision. With Hollybridge arguing one way, and then on the other side of it, of the camp. You've got people saying it's the pulvina, and then at some point there was this paper saying that actually whether it's the pulvina or the LGN depends at which point in time the lesion was. And that was based on a study in humans. And so, yeah, and also work that we've recently done, we showed that the pulvina pathway seems to enable blind sight in patients with projections to STS. So I'm just wondering, do you have any data or intention to look at pulvina as well?
There's two parts to my answer. The first one is that I think it's too simplistic to look at it's one or the other, because the different, like affective blind sight, for example, is something that people link to the amygdala, and you worked on that, there's different circuits. So also, depending, blind sight is, well, the definition would be that it's the subconscious abilities, whereas there's also things that the person can sort of vaguely, consciously perceive, like motion. So to me, the most likely scenario is that there's a number of those smaller circuits that survive, and they each contribute. And depending on what exactly a person, because remember, here we can create precise lesions that are more or less the same. In humans, you don't have this luxury, right? So it's what survives will depend also on that. Now, in terms of the pulvina, the reference actually was a study done in marmosets by James Bourne's group. What they showed was that when you're born, you have a larger projection from the retina to the pulvina that then goes to the extrastriate cortex. And over the first few months of life, this projection is trimmed and becomes much smaller. Right? So if the lesion is made early enough, that pathway seems to be spared. And that could create another way why, because, we know clinically that there are, if the lesion is early in life, the amount of abilities that are surviving is much larger, right? So, for example, people might they might have hypoxic damage to the posterior pathway early in life or during birth, and then leave more or less unaffected until they have to go to school, and they can see things very well, and the teacher refers to the ophthalmologist, and the ophthalmologist say, yes, you have damage to V1, you never knew that, right? So I think that it's clear that there's an age effect, and in the case of the LGN, we didn't find dramatic differences between ages, so that to me would be meaning that probably it's one of those abilities that survive whether you have the lesion in adulthood or very early in life.
Thanks.
So last question.
Thank you. Hi, fantastic talk. No? Okay, I can continue. So, when you see more empty projected neurons after the lesion, do you think those neurons are forming new synaptic connection, or it's rather there might be some existing connection, but they will be like very weak, and then typically, normally you would not see that with retrograde tracing?
This is my current research. All of those hypotheses are being tested, including generation of new neurons. Right. In fact, you have your reactive neurogenesis in the LGN. So new neurons appear in the LGN only on the side of the lesion, but we haven't found yet that they project to RMT. So at the moment, we are leaning towards the explanation that there are connections that exist there. They exist to the cortex, and they just branch horizontally in the cortex to more effectively.
So they will generate new branches.
Yeah. So existing connections, so that could, right, create more synapses in the cortex. So that's currently, but as I said, we don't know.
Yeah, but that's cool. Thank you.
Thank you very much again for Marcello.